From Microbe TV, this is TWiEVO, This Week in Evolution, episode number 51, recorded on January 21st, 2020. I'm Vincent Racaniello, and you're listening to the podcast on the biology behind what makes us tick. Joining me today from Salt Lake City, Utah, Nels Elde.
Hey, Vincent. Great to be here from Elde Lab Studios, and Happy New Year.
You too. This is our first of 2020, right?
Yeah, we're moving forward.
Yeah, Happy New Year, listeners.
Correct.
Another year of TWiEVO.
Well, in the fifth season.
The fifth season, right? That's
Yep.
cool. We could do seasons.
Uh-huh.
If you like what we do here at TWiEVO and all the other podcasts, microbe.tv/contribute, consider supporting us. More on that later. Got a little email here from Martin who said, thought you and Nels might want to mention the passing of David Hogness in your next TWiEVO. David Hogness, quite an amazing scientist.
Agree. One of the giants in the field.
Mm-hmm. Biochemist, geneticist, developmental biologist at Stanford. I did not know him, nor did I ever meet him.
Yeah, same here. I knew of him, of course, and all of the groundbreaking work and folks who've emerged from his lab. Kind of a who's who list.
Yes. Steve Goff was telling me he's trained amazing people.
Yeah, exactly.
Article in The Scientist, it says he brought molecular biology to Drosophila, discovered the first core promoter element in eukaryotes, cloned the Hox genes. Keeps going.
Yep. Tata box, the old famous
David Hogness.
Transcriptional.
Yeah, Goff said he gave a talk once and he presaged everything about the recombinant DNA revolution.
Yeah, interesting.
And before anybody had done anything, he said, this is what's going to happen.
Yeah. Also really interesting. So I think I had read that news piece in The Scientist a week ago or whenever this came out. And, or actually, I think it was the description from Stanford University put out a release because I think he was working into his 90s just before he passed away. And one of the interesting things they noted as well is actually how rarely he published stuff.
Yes.
Right. And I mean, in some ways, I feel like today's science almost demands, right, this publish or perish idea as you're being evaluated on things like tenure, stepping through sort of the hoops of academics. But really, I think fun to remember that not one size fits all in science, and that, really working deeply on these big questions, these hard problems, it is also a complementary part of really pushing science forward. And so I thought that was just a nice reminder of like not getting kind of caught up in the sort of business today and of science today, but to think about how science moves forward sort of from a larger perspective.
Yeah, it's true. We don't tolerate much diversity in science these days in terms of people having to just do the publish perish thing.
Yeah.
I asked Goff about that, and he said he got support from his colleagues in the department. There were enough people with support, and they gave him some money each. And can you imagine? Nowadays, nobody would give me money.
Kind of crowdfunding within your department?
Yeah, within your department. Of course, I crowdfunded outside of the department that worked, but not enough to sustain a laboratory,
Yeah.
I've asked some colleagues for support in the recent past, and I have not gotten it. Everybody's got their own issues, right?
Well, and we've moved, just as inflation has moved forward, a lot of science funding hasn't. And so if we just even consider just the core R01 mechanism and the modular budgets at $250K, I mean, you can do the kind of reverse economics on that and the math and sort of the core amount of funding versus how people are scrambling today. It's actually pretty drastic. Where I think as experimental scientists, we've sort of become like the frogs in the boiling pot of water where it keeps getting warmer and warmer, but so gradually you don't notice kind of energy. So, yeah.
I think other countries recognize that science deserves more funding. I think China is surpassing us in science funding.
Indeed.
They recognize that it's important for not only a healthy populace, but just understanding the world around us.
Yep.
There is a lot to be said for that. So anyway, it's good that he was able to do so much. And Box, next time you hear Box, he discovered that. Colony hybridization, which many of us have used to isolate bacterial colonies containing certain DNA sequences, another one. Amazing.
Yeah, and that transition from sort of the molecular biology, earlier genetics era into genomes, that was, again, one of those things that almost seems, we almost talk in genomes these days somehow.
Yeah, for sure.
Remembering that transition.
Thank you, Martin, for pointing that out.
Yeah, great.
I missed it, actually. It happened. He died on December 24th.
Mm-hmm. Yeah, I saw that release from Stanford. Nice idea to lift that up.
What do you have today for us, Nels?
Yeah, I'm excited, Vincent. So, most of our TWiEVO episodes have been out, so to speak, at the tips of the branches of the tree of life, right? We've been talking a lot about, whether it's virus evolution, other microbes, or rapid evolution in different settings, where it's kind of the blink of an eye of evolution. One of the reasons I would say TWiEVO, we focused on that, is it kind of fits into our some of our core principles or ideas that how can we lift up work that considers both sort of the evolutionary ideas, but puts it into some sort of experimental framework, or uses the emerging technology, the sort of genomic era or post-genomic era, however you want to describe it, to be able to really kind of understand old evolutionary questions in kind of new ways, or find that confluence between fields. So almost as a side effect or byproduct of that, we've sort of, I think, ignored deeper evolution, sort of. So certainly we haven't talked much about things like the origins of life. We haven't talked much about sort of notions of an RNA world before cells existed, or how do you sort of begin to transmit information through generations? How does heredity get set up? All of these things. And so today we'll be exploring one facet of this deeper evolution. And this is in regard to sort of early protein evolution. So we're probably not at the very beginning of the origins of life, but somewhere pretty close. And thinking about how do just chains of amino acids, or really simple peptides that might even just show up in the absence of life at all, might start to come together or assemble in ways that they would take on sort of biologically meaningful or useful properties as ancient proteins. And so the paper or preprint in this case, is entitled Primordial Emergence of a Nucleic Acid Binding Protein via Phase Separation in Statistical Ornithine to Arginine Conversion.
Cool stuff.
Yeah, kind of fun.
Yeah.
And, a little bit of a preview. I think we'll be getting into some really cool ideas. I think the work will also illustrate some of the challenges of really kind of not only just wrapping your brain around how things might have been, but then coming up with sort of plausible scenarios to put forward. Of course, now that we're considering something that happened, 4 billion plus years ago or somewhere in that neighborhood, there's it's really tough to get sort of validation in a lot of ways. And so are the authors building something like a circumstantial case for sort of what might be possible?
More than 400 million.
Yeah, 4.5 billion.
Billion. 4 billion, yeah, for sure.
Maybe more than 4 billion.
Yeah, for sure. Well, the thing is that at one point on Earth, there were no proteins. There were just RNAs or something. Probably just RNA, right?
Yeah, so that's one. Yeah, so there's this idea that I think has gained, certainly quite a bit of attention, and even out beyond sort of science circles, of the RNA world. And one of the big drivers of this was work by Tom Cech and others in the discovery of ribozymes. So these are RNAs that actually can be catalytically active, right? Both encoding the nucleic acids so that you could imagine passing or replicating these so that you could pass that information on. But then also encoded within that information is actually a biological function, the enzymatic or ribozyme function. And so I think that idea has really dominated in origins of life thinking, that you can start to imagine putting those together because the RNA is there. Things like those nucleic acids you can find in abiotic scenarios outside of any life. And so that could give you something like a plausible trajectory to start building your way. Once RNA gets organized and then compartmentalized, then you could start to move into a ribosome-based translation system where you start to bring in the peptides or the proteins that do most of the heavy lifting of enzymology today.
Could we just short circuit and have someone just give us a book on all that happened?
Yeah, good. There's several books out there. There's also some labs that are doing really great stuff in addition to the lab we'll be talking about today. So John Yin is at University of Wisconsin at Madison, at the Morgridge Institute of Sciences there. Also Jack Shostak, who probably well known, famous for his Nobel Prize in his part in the telomerase, discovery of telomerase, the ends of chromosomes, and how do you solve the DNA end problem for chromosomes. And then we'll also add, of course, so today's the lab from today's author. So this is Dan Tawfik's lab in the Department of Molecular Science at the Weizmann Institute in Israel. It's really fun too. If you go to his website, I like the branding here: protein archaeology to synthetic biology.
Now you have to brand your lab, huh?
Yeah, that's right. I think we'll see some of both actually in this paper.
I would point out also that there are people working on this idea that there was an RNA world and nucleotides arose that could assemble into longer chains and then could copy themselves in the absence of any enzymes.
Yeah.
Right? Yep. Cleavage, yes, as you've mentioned, self-cleaving, and there are people who do this chemistry. They make various nucleoside-looking compounds, and they can get them to assemble and copy themselves. And it's really, maybe some point we should do one of those because it's really cool stuff.
Oh, absolutely. And, if any of our listeners have recommendations on kind of origins of life books, that would be great. I'll confess that I've haven't dug deep into that area. I spend most of my scientific time out on the tips of the branches of the tree of life.
Yeah, sure. Yeah. Most of us do.
Yeah.
It's hard to get back there, but it's fascinating to think of where things
really
started.
Yeah. And I think, when done well, which is a challenge, I mean, the courage and the energy it takes to sort of put this, I think it's really exciting and fun to consider, which makes the paper a fun one to go through. So I guess, go ahead.
Set the stage for us. So we have a transition we have RNA and then we're getting into proteins. So this is a primordial world, and tell us a little bit what we think is going on there.
Yeah. And so I think, how the RNA world, those ideas fit into it, this might be coming kind of a little after that, might be one way to think about it. But, whether these things are kind of happening in parallel or if one has to come before another, RNA before protein, all those things, I think either could work. I would say the way this paper sets up is to ask the question, how do ancient peptides, which would be really like short strings of amino acids, come together and grow up, in a sense, evolutionarily speaking, to become biologically useful proteins? So more complexity. And then at some point, you go from just what you might consider abiotic amino acids. And so this would be, amino acids that could chemically arise without sort of a biological form around it. So things that we would recover just in the environment, even today, or on meteorites or coming out of volcanoes, things like that. And so you have you might consider it a limited alphabet. So as we all know, today we have 20 amino acids that show up in our proteins. They charge tRNAs and then are part of, show up at the same time as messenger RNAs at the ribosome to get assembled or covalently linked into peptides and into proteins. But this is, so we're before, the paper is setting us kind of before that would happen, and in the primordial soup. So these things are just coming together. And then how do you start to transition over these long time frames into proteins that actually fold up and have sort of active properties?
Now, the primordial amino acids that we think there were fewer, right? They're not 20.
Correct. Yeah. And I think this is based on the fact that just what can you find out there in the environment, right? So you're like a geologist would find these, or, just as you're doing kind of chemical analysis of mud puddles or things like that, yeah, you'll find, I can't remember the exact number. It might be something like 10 of the 20 amino acids are abiotic in the sense that that you can just find them outside of a biological system.
There's some that still exist, but I guess they all exist, right? But they're not coded for by triplets today.
Yeah, exactly. Yeah. Right. Yeah. Yeah. And the authors nicely get into that point at the end. So in the title, they mention this ornithine to arginine conversion. And so arginine is one of the biotic amino acids that we're pretty familiar with from our biochemistry classes. Ornithine is not. So this is an abiotic, and that kind of has a fun starring role later in the paper.
Mm-hmm.
So to begin with, I mean, it's kind of a big question, right? So how do ancient peptides, abiotic peptides, grow up to be biological proteins from an evolutionary standpoint? So, and this applies to a lot. So I think they actually chose a pretty compelling system, an interesting model to do this. And this was the property of double-strand DNA binding, which many modern proteins do. And this was, you can imagine that this is also a pretty fundamental and sort of evolutionarily formative biological property that could kind of really spark the complexity of life around us, because now you're starting to put together systems to not only, have nucleic acids around, but to actually work with them, right? To develop or to almost build scaffolds or systems that help to conduct biology. So we're talking about, DNA binding is important for things like transcription factors. So David Hogness, in the fundamental work that he did in his career, how do transcription factors recognize DNA and different motifs to go from that information that's found in nucleic acids to actually delivering on a function through a messenger RNA or intermediate that then becomes a protein. I'd also mentioned self versus non-self recognition, right? So how do we tell our own nucleic acids, DNA, from one of an invader, from viral DNA, for example? And we have a number of DNA binding proteins kind of patrolling the cytoplasm and trying to sound the alarm if there's something in there that's trying to take over our own cellular machinery. Genome organization, right? The histones. These, like, how do you pack up a chromosome? How do you pack up DNA into a chromosome, into a massive genome, and are able to either tolerate billions of base pairs or gain, kind of concentrate information in them? So, yeah, I think a good choice of both a measurable phenotype. DNA binding is pretty easy to measure or observe, but then also something that sort of connects in a deep way to our biology.
They also say that these DNA binding proteins are pre-LUCA. What is that, like olive oil or something?
I love it. Yep. LUCA, the last universal common ancestor. Yeah. And so that kind of gets at, so pre-LUCA, right? So this is this idea that we're trying to, or the authors are trying to consider even before what we would consider sort of the very first life form, the first cells that were coming online, that they're imagining that these are some of these fundamental properties would have to emerge from outside of biology to somehow contribute to the beginnings of it.
So we know that these DNA binding proteins were around pre-LUCA, right?
Yeah, that's the idea, and I think it might follow logically, just as you. But yeah, I don't know how to judge the evidence for that as being airtight, but it might just be just kind of Gedanken experiment, so to speak. And just as a disclaimer too, again, there's no time machine, right, that we could go back to billion years, and so we end up depending on some of the really, this is before fossils, right, and so we're like making all these massive inferences based on the dating of rocks and the chemical compositions of them, trying to borrow from observations at like deep sea vents to try to think about what were the kind of chemicals that were colliding in interesting ways. Really, a really long time ago, like beyond the grasp, at least of my brain, to even conceptualize how it's a billion years to a human brain. It's crazy. So anyway, so that's a challenge, right? Is to start from the beginning. And so in fact, so the authors want to set out to kind of provide what they're describing as an experimental trajectory, or a trajectory that they could validate through experiments, of how, you might start with a really simple polypeptide, again, made from abiotic amino acids. You can find them lying around in nature. And how would this then transition or undergo a trajectory to becoming a modern protein? Mm-hmm. And so I think what they do is pretty clever, and it kind of makes sense, is they actually start the other way around. So you start at the tips of the branches of the tree with modern proteins, in this case, modern double-strand DNA binding proteins, and use those modern ones and comparisons of them to start to work your way backwards. And so this has actually kind of come into, I would say, in evolutionary genetics. This has come into fashion, and a number of labs spend a considerable amount of time doing phylogenetic analysis of extant existing proteins. And you can do this from highly diverged paralogs, different families, but you're looking for sort of kind of a kernel or a foundation of what's the same over these long, if you take really unrelated modern species, what are the amino acids that are still common to them? Mm-hmm. And that conservation, that deep conservation, gives you a sense that that's been around for a really long time. It's ancient. And then there have been some advances, I would say, in the last decade or so in ancestral reconstruction. So using statistical methods, usually maximum likelihood, to actually predict the sequence of the ancient proteins, in this case, the ancient DNA binding proteins. And there, so if you've got, let's say you take every species that you can get the gene sequence, and the authors will be trimming from full proteins to just motifs or shorter peptides to begin with. But if you took every single sample you could get, and if they were 100% identical, then your ancestral reconstruction would be easy. It would just be today's sequence. It hadn't changed. But inevitably what happens, even in sort of highly conserved situations, is mutations happen, and they're tolerated. And so then, in a sense, that kind of throws like sand in the gears of trying to figure out what did that ancient protein look like. And so evolutionary biologists have sort of fallen back on statistical arguments for, but given this arrangement, and here's the most probable amino acid at each position in the ancient protein. I would put like a slight caution on that, or slight grain of salt on that, and that these are predictions, and we're starting to then make predictions at each site, put them together into a longer stretch. Just your probability of getting everything exactly right goes down pretty quickly. Mm-hmm. And so predicting sequences is simple. You can get a computer to do it, crunch through it in seconds. However, actually knowing what was really there back a long time ago, not nearly as easy.
Mm-hmm. Sure.
Okay. So anyway, they decide to focus on this motif, the hairpin helix motif, or Triple H, might be a quicker way of saying it. It's a 4-helix bundle. If we look at Figure 1A, they have a nice little illustration of the protein or the peptide fold. And as it's addressing, it's on the left, kind of in cyan, and as it's addressing double-strand DNA on the right. And so I think it's about 60 or 64 amino acids, something like that. And really highly conserved overall. And actually, you can get a sense of that. So if you look in Figure 1C, they have this thing called the logo diagram. And so they took the helix hairpin helix Triple H motif from as many, or sampling, for kind of ancient family, or for really well conserved families. And then the logo, the way that works is it shows the letters, the actual amino acid, and how common they are. And then you just stack them on top of each other. And so you can see for about half of the peptide, it's almost just one letter. And that's really deep conservation across the entire family. And it's ranging from bacteria to humans, basically. So that's gives you both a sense of that this is ancient and that it's hasn't changed a lot. However, there are plenty of cases where there's actually 5 or 6 amino acids that are stacking in one position, and they're sort of different sizes. And so that's where it becomes really difficult, even using kind of the most sophisticated ancestral reconstructions, to know really what did the ancestor look like. Mm. And so, yeah.
And so these 2 helices, right? Mm-hmm. They're separated by a hairpin, which allows them to bend. And the two helices contact DNA.
Yeah.
And they have to be basic amino acids, right? Because the DNA is negatively charged.
Exactly. Yeah. And it's a really good point, and that's going to come into play here in a few minutes. So the first thing they did, or the first observation they made when they do the ancestor, and what they noticed, which is kind of cool, is that compared to all of the modern examples of these Triple H motifs, this one the ancestor looks more symmetrical. Meaning, the first half looks a lot like the second half.
And by the way, they made this, they synthesized it chemically.
Good point.
And it's soluble, which is good because, right?
Yeah. And good point. And by the way, so when you're doing these ancestral reconstructions, and this sort of helps give it a little bit of weight, is that you'd imagine a lot of combinations just wouldn't even become they wouldn't fold up, they wouldn't be soluble. Yeah. You would just make junk.
Yeah, this one seems to fold, and it binds DNA too. They can do a DNA binding assay. So
yeah, exactly.
Great.
And so already, is it the precise ancestor? I don't know. But does it fold? Does it have some activity? Yes. Yeah. And so it's already kind of cool, I would say. Yep. And then,
so what's this symmetry business? Why is that cool?
Yeah. So the reason they put that forward as being cool is that it might reflect that there actually was an ancient duplication, right?
Yeah,
so
right.
And that you're kind of reversing the clock a little bit and getting back to closer to those the deeper evolutionary past for this thing. And in fact, they go on to try to make it perfectly symmetric, to try to rewind the tape even further, so to speak. And so when they do that, they found out, actually, no, you can't push it that far. Now it doesn't bind double-strand DNA anymore, at least for the half peptide that they chose. That didn't work. And so I think it's worth it to just kind of, as you go through, to always be considering kind of alternate hypotheses. Well, maybe their symmetrical ancestor didn't work, but maybe the actual symmetrical one, there could be all these different pathways. And then in the end, I think, what makes this for me really cool is just as they step through the kind of trajectory, I mean, it is plausible, right, in some way. And so is it the exact way history happened? No, probably not. But is it a way that it could have? And what's the value of thinking about things that way? I think that's what's really fascinating about this. Okay, so anyway, they're actually just getting started. So they still want to keep trying to step backwards in evolutionary time, in a sense, to kind of reverse the trajectory. And so what they did was kind of, almost by trial and error. So they did that perfectly symmetrical. It didn't bind double-strand DNA. So then they say, okay, well, hold on. Well, let's make a compromise and put one of the extant linkers into the so it's still perfectly symmetric, but now they put a linker to try to use, I think, a little bit, like just protein engineering principles to do something that might kind of restore that. And in fact, when they made that compromise by putting in a linker, short sequence, just a few amino acids, now all of a sudden it binds double-strand DNA again. And so they decided to use that as their basis. So they felt like having it symmetrical to recreate a potential exact duplication was worth it above sort of the half ancestor, which was more symmetric, but not completely. And so that's the one they use to go forward to try to make it even more primordial. And so they note at that stage that in what they're calling their symmetric Triple H, that it had 13 amino acids. And they wanted to get even more primordial, meaning that they wanted to only include abiotic amino acids.
Of which there are 10, right?
Correct. And so they wanted to get rid of histidines, phenylalanines, and tyrosines, which are biotic. And so then, what's your building list? And so they're trying to find amino acids with similar properties. And that, I guess, is relatively straightforward for phenylalanine and tyrosine. But histidine, not so much. You could change to lysine, but that's not abiotic. But they want the challenge here, and you pointed this out, Vincent, was the basic nature, right? So to be able to bind to negatively charged DNA, you want to retain basic residues to facilitate that binding. And so they turn to arginine, again, not abiotic in this case, but they're kind of, going stepwise. And so I think they remove a handful, like maybe a dozen or so of the histidines, phenylalanines, and tyrosines, and then put in arginine instead. They called this one Primordial Triple H arginine, and that worked. So they're down now to I guess 9 primordial amino acids plus the arginine.
Right.
So getting closer. But then this was kind of cool. So I don't, a little bit of a curveball for me, at least. I don't think a lot about amino acid and protein synthesis. But so they turned to ornithine, which is an abiotic amino acid. I guess it emerges if you do volcanic spark discharge experiments. Have you done anything like that in your?
No, we don't do that. No.
It's not my jam either.
We don't do meteorites either.
And you can also find ornithine in meteorites, I guess, if you're just sampling the chemicals. And so based on its properties, basic and
it's basic, yeah, that's
and being abiotic amino acid, they decided to go after that one. And so now we're getting into, I think, some pretty heavy lifting when it comes to the chemical synthesis to add.
For sure.
And so I think they go through cysteines that are post-translationally modified into selenocysteines, which somehow give you a handle to then swap in the R group for ornithine, which, I don't know, to me sounds pretty cool. And they end up with what they're now calling the primordial, or I guess they called the other one that, the primordial HHH orn, or for ornithine, where it's now all abiotics.
Well, 14 arginines are now ornithine, right?
Yeah, exactly. And by the way, that's a lot. So when it had 14 arginines, that's like a quarter of the amino acids, right? Mm-hmm. It really illustrates, I think, the basic nature of this to be a DNA binder. And so, but what they found was this guy only really weakly binds to DNA, and the peptide itself, or the protein itself, is basically unfolded. So it's a little bit of a mess. But the fact that it is working even a little bit, they're pretty excited about. And I guess, in some ways, maybe that feeds somewhat intuitively into our thoughts about, like, something that's just emerging from the primordial soup is probably pretty inefficient and weak.
Yeah, it's the way it starts, right? It's just random stuff coming out with maybe a little bit of binding, and then it goes on. Yeah, it's just probably how it worked. But then I guess the question becomes, like, how many kind of non-working things did you have to try, or were tried just by trial and error, or by chance, random kind of processes to get something that works? They give a number for that.
Oh yeah,
you remember?
Oh, right, yeah.
So it says here, just the function, the likelihood that you're going to get by randomness some function. 10 to the minus 11 for binding nucleotides.
Yeah, and I think that, if I'm remembering right, that's even for just like using out of the box like regular
that's right.
amino acids, right? And so now, take that number and I think make it even smaller or less probable. Yeah. If you're now adding, you can throw in abiotics here or some combo of who knows what that went on. And so on the one hand, I think it's really cool kind of intuitively, but on the other hand, it just illustrates to me, like, when you're making kind of junk, how many junky things, the signal to noise feels like it becomes an issue to me in terms of, like, is this really happen? But again, really provocative, interesting. And also the fact that they started kind of from today and worked backwards to get here, I think is also kind of a cool approach.
So we have a little protein, a DNA binding protein. It's made up of abiotic amino acids, right? And it doesn't fold, but it does weakly bind DNA. That's what we got so far.
That's what we got. And so I think that kind of, in a sense, that's how far they rewind the tape. There's a little twist to this that'll come up in a minute. But then what's kind of interesting is now they kind of go back forward again. So there's this natural process of guanidination, which starts to slowly convert the ornithines into arginines, actually. And I think this is in the title, they mentioned the statistical conversion. And I think what that reflects is just that you're now making a population, or there's at some frequency or at some probability it will start to convert.
So initially a few residues, and then more and more as they push the reaction, I guess, right?
Yeah, that's right. And so so then the idea would be that, like, as time unfolds, and I guess even kind of, relatively quickly in this sense, that you would start converting to arginines, and guess what? You get better DNA binding in a case like that. Oh, actually, that reminds me. So they do try to go back even a little bit deeper, because one of the sort of, pivotal events they're hypothesizing is that duplication happened, right? They spent all of this time kind of messing around with symmetry. And so then they decide, well, let's just chop this thing in half. So it's not symmetric anymore. It's just sort of, right? And you're down to 30 amino acids. And what they found is that actually that thing didn't bind DNA. And so that's another kind of point for why they thought the duplication sort of potentially going from ornithine to arginine through that guanidination conversion, and then that the duplication was really important.
Now, when we, in all of TWiEVO so far, when you and I talk about duplication, we're talking about at the nucleic acid level as the driver, right? But that's not the case here, right?
Well, yeah, so that's a good point. And that's right. And I don't know that, so my sense, because we're really deep in evolutionary time here, before you would have sort of that nucleic acid template.
Exactly.
Yeah. And so how does the propagation work here? Is this, so you could certainly imagine short peptides coming together, and there could be a covalent linkage that could lead to a peptide duplication. But then is this getting templated almost like a prion where you've got protein inherited?
It might.
Yeah.
It has to be. But the thing is, okay, you get this this original sequence, and you duplicate it. Now it's weakly binding, but there's no DNA around, so there's no selection to make it better, right? Not yet, anyway. Maybe that happens later.
No, I think that's right. And then that's one problem, not a trivial one. And then the other problem is if this is all happening, out in like mud puddles or under the sea or in really dilute environments, then how is it that you bring these things together in a concentrated enough form where you have enough collisions happening that you can actually select on this?
Exactly.
And so that's kind of a little bit of the cool, or like the second shoe dropping for this study, is that they consider that key property of how do you concentrate these biological reactions that would be needed for life.
Yeah.
And so what they found is when they mix any of their primordial peptides, I think even the non-duplicated, the duplicated, if they add poly-U, so nucleic acid, just a string of U's in a row, then some magic happens. You get these coacervates.
Mm-hmm.
Yep.
Is that some kind of a cocktail? No, that's ceviche. No, I wasn't familiar with coacervates at all.
Yeah.
The hell are they?
So these are droplets where, and actually this has become really almost like a massive, more than a cottage industry in cell biology. There's this idea of phase transitions or phase separations, where you have basically molecule-rich droplets that then phase separate from like dilute situations. Coacervation, I think, is a older sort of term for these colloidal droplets. So you have two different liquid phases that coexist. One is dense and polymer rich, and that would be, where you would expect these proteins to be interacting with nucleic acids, for example. And those would sort of be almost like a lava lamp. That would be the wax kind of bubbles within the dilute stuff around it. And so that idea is old. And in origin of life research, some of the original proposals, so I guess this is a guy, Oparin and Haldane, and this is JBS Haldane, who's well known in sort of evolution genetics, population genetics circles for a lot of his work and on the sort of the math of inheritance. But he was also thinking about some of his science buddies, some of these earlier questions about origins of life. And so they put that idea forward. This must be, 70 years ago or more, something like that, 50, 60, 70. And here it looks like that's popping up in kind of a cool way in this paper when they bring the really simple nucleic acids together with the really simple or proposed primordial proteins, that you have that property. And so it's almost like two birds with one stone, where you have now like some interesting potential biology that is getting concentrated at a level where you can imagine now maybe natural selection being able to act because there's things coming together or there are compartments forming. And so that's that's kind of the idea that they're putting forward with what they're proposing in reverse order now. If we kind of flip the script, you would start with these really simple peptides, all abiotic, limited alphabet. You'd add a little bit of nucleic acid. In this case, it's just a homopolymer of U's, and then you start somehow marching through the steps where then you duplicate the peptide. It starts to diversify. You'd then have to somehow be in a ribosomal setting. So the reason they claim that, so one question that would kind of obviously emerge from that is, well, why was ornithine lost? If that's so great for being there at the right place at the right time and starting to bring in the chemical properties that would allow for the basic properties that would allow for DNA binding. Why was that lost? And I think it turns out there's a chemistry reason behind that. It's hard to charge a tRNA with the ornithine, and so
right. But so I guess arginine is an improvement, right? Yeah,
and so that would be part of that, trajectory, so to speak, stepping forward, and then the idea would be you kind of continue on. All of this stuff gets taken over by ribosomes, and now you do have a genome. Might be DNA. Somehow you're going through, the central dogma, DNA to RNA to protein. And then we get to the tips of the branches of the tree where they actually started their experiments with all of these Triple H motifs that you find in these diversified families of proteins that have the properties of binding double-stranded DNA.
This is just emphasizes that this all happened in tiny, tiny steps, really, over really long, long periods of time, right?
Oh, man, yeah.
Didn't all happen at once. Everything came together, boom. You had that first cell. It just goes really slowly, and stuff may have emerged that didn't get used for a long time, right?
Yeah. Oh, yeah, for sure. And there's actually some basic building blocks here that I'm not mentioning. So, like, where do the membranes show up, right? So you've got maybe this phase separation of these droplets, but then how do you become membrane-bound compartments, right? The first protocells. And that was some of this thinking about the coacervates or coacervation.
Yeah.
They were trying to put forward a little bit of that protocell idea. How do you go from phase separation to then thinking about like actually adding sort of biological membranes around there as well. So
yeah, there is a good literature about all of that. What was the guy's name? I'm reading his books lately. Let's see. It is Oxygen book. Let me see.
Oh, is this Nick Lane?
Nick Lane.
Yep.
Yeah, and in several of his books, he talks about how cells emerged and where that might have happened, and there's a whole literature. He's one of many, many people who are really interested in this.
It's fascinating to think about.
Fascinating. Everything we know has a proton gradient, right? That drives life.
Yeah, right.
So that seems to be a key early event, and many people think it happened at these tall things at the bottom of oceans, not vents, but I forgot what else it is. It's something else.
Smokers or something?
No, the smokers are not the right place. Some people think, but it's not right because the other structures whose name is escaping me, they have water coming up from beneath the crust and then the ocean water, and basically that gives you a proton gradient.
Oh, right. Yeah.
And these things are divided into little cells. They grow and fall periodically because they're made and then they dissolve and fall down. But he and others have. So if you're interested, go look at his books. I found it fascinating.
Yeah, good writer, Nick Lane, really interesting guy, and practicing, biochemist as well.
Yeah, as well. Yeah.
Yeah. No, and so at least from where I'm sitting, I mean, what I really enjoy about the kind of work like this is it kind of, starts to put at least a scaffold or outline into what's possible or what could happen.
Yeah.
I think if you hold to it too tightly, that then, it becomes problematic because just like you're saying, so this is all happening, all this kind of complicated stuff is happening over the course of a really long time, billions of years. And so, just even kind of the tempo of it or what has to happen in the right order to then sort of actually get towards Luca It seems like there's so many potential pathways.
For sure.
But that's what's really exciting is to kind of, in this case, kind of working backwards to get to something, and then just kind of considering what might even be possible to begin with. Yeah.
I mean, if you have, let's say, assume we have RNA first, right? And then these peptide coalesces are changing over time. And then one comes that combine the RNA, maybe it gives it a different property, right?
Mm-hmm.
Of some sort, stability or the ability to be longer. It could happen at that level. It doesn't have to control gene expression right away, right?
Yeah, no, of course. Yeah, no, and almost I think would have to. And that's one of the, I think fascinating things about evolution too, is just the convergence or the independent evolution of like the same properties in different times or different places.
Yeah.
And so something that worked out, something that bound to like an RNA, like you're saying, that somehow stabilized something, could then hundreds of millions of years later, sort of that same property might perk up again, but for another function altogether.
Yeah, absolutely.
Yeah. And so those themes, that idea of independence or convergence, that is really a great molecular beacon or a beacon to figuring out like the things that keep working over and over. Those are probably things that were important or useful. Somehow, through the trial and error.
I agree. I think you have to step back and not think of today's function, but you have to step back and think, oh, well, I might have done something else. And eventually got around to doing this, but something, because it's too complicated for it to be originally a DNA binding protein, right? First of all, there's no DNA and there's no promoters.
Yeah.
So, but they're there, the peptides are there. So you have to find a reason for them to persist and think of it differently. And also you have to break it up into billions of steps,
Oh, man.
Obviously.
Yeah.
I've learned that from TWiEVO, actually, that the only way to deal with it, to wrap your head around it, is to break it up into steps like this one and look at them one at a time.
Yeah.
Otherwise it's overwhelming. You go, how the heck could this have evolved? Right.
Yeah, that's right. Yeah. Well, and that's where you can fall into some traps. So, the idea of irreducible complexity. Mm-hmm. How is it that we could have a modern eye, that, you can't just, it's not easy to see that in one step, just like you're saying.
Sure,
but then as you start to break it down and look at comparisons to other sort of solutions to light sensing, now all of a sudden you can start to, at the very least, imagine trajectories, experimental trajectories or kind of biological trajectories where all this complexity could actually emerge. Yep.
That's one of the things Lane goes through in one of his books, The Eye, because he said, people figured couldn't have evolved. And he said, no, here's why we can see all these organisms that exist today at various stages on the way to a current modern eye, right?
Yeah.
It's really compelling. Yeah. I like this paper a lot. I think it was cool.
Yeah, I agree. Pretty fun. And it's good. So that's, one of our goals, of course, on TWiEVO is to sort of shop around into different parts of the field. And so I think we were overdue for going a little bit deeper and keep our eyes open for more sort of explorations like this, right? Yeah. Cool.
All right. Let's do a couple of email.
Yeah, great idea. So Anthony writes, though sex determination seems to be fixed at conception by chromosomes, that's far from so in many other species. Clownfish, seen in most any aquarium store, use sequential hermaphrodism. And that's just one of many strategies. The link to a paper, PLOS Biology paper, describing clownfish that actually, depending on, how many other clownfish are nearby, you'll have a male and a bunch of females. If that male is eaten or lost or dead, then now one of the females will step up and become a male. It's a really cool example of flexibility of sex determination. Beyond just sort of the chromosomes that you were born with.
Nice figure in the paper of showing how it works among different species.
Yeah, that's a cool reference. We'll put that in to the show notes here. So he says, there was a mention on TWiEVO 50 of hermaphrodites and self-fertilization. This is not always so. Earthworms are hermaphrodites, but still require a partner. Thank you.
So I looked this up. It's called simultaneous hermaphrodites.
Yeah.
So sometimes, depending on the species, they may or may not self-fertilize. You may have seen a picture of two worms fertilizing.
Oh, yeah.
And, they're kind of stuck together, and that makes sense because I guess it would be hard to self-fertilize just physically. But so earthworms have both ovaries and testes, but they have a protective mechanism against self-fertilization. Mm-hmm. And it occurs when two worms meet. So that's interesting to have a protective mechanism, because there's obviously a downside to it's cloning yourself, and that's not as good, right?
That's right. Yeah. Yeah. So that's, it's good to have some options. Like,
I suppose.
So that's true. C. elegans is another case. The nematodes, microscopic worms that often are hanging out as hermaphrodites, but there still are males. And you can have really cool sperm competition. There's a lab just down the hall from me at Utah, Jillian Stanfield's lab. They're working on this. And so having sort of the ability, maybe it's, one way I think about it is a little bit of a balancing act, where depending on your environment, there are times where you just want more, or there are times when reproduction as clones, as a short-term strategy, is pretty successful. But then sex, for all of its downfalls, including losing half of your gametes, half of your genetic material to your partner, has still proved to be pretty diverse, useful, and persistent as a strategy. And so, why not do a little of both in some of those cases? Yeah.
So Anthony also has another one here. He had written to Katie Peichel, right?
Oh, great. Yeah. Yep.
So it's our paper last time, and I'm dreaming of a Y chromosome.
That's right. We were talking about stickleback sex chromosome evolution.
So he wrote to her and said, through Professors Elde and Racaniello's TWiEVO, I had the good fortune of hearing of your paper. And she's probably thinking, what the hell is this TWiEVO?
What have we done?
Before the evolution of the Y chromosome that you describe, is it known, assumed that the method of sex determination was sequential hermaphrodism, as in clownfish? Is there a fish that shares a last common ancestor with the three-spined stickleback that uses a method of sex determination other than XY? If yes, can it be assumed that the ancestor of the sticklebacks used that method, or would it be that the ancestor used XY, sticklebacks changed from that, and then the three-spined switched back to it? And then she answered, Dear Anthony, thank you for your interest in our work. In fish, the mechanisms of sex determination is very labile and switches quite rapidly. Thus, it's impossible to determine what the ancestral state is in fish. Even in sticklebacks, we see that the sex determination mechanism and sex chromosome differs between closely related species, so we do not know what the ancestral mechanism is in sticklebacks. And she sent a couple of papers to him to read about.
That's cool. Thanks, Katie, for weighing in on that. Katie and I had a nice email exchange as well when she heard our podcast. So I overlapped a bit. So Katie was running her lab at the Fred Hutch Cancer Research Center while I was a postdoc. And then after I left, she left for Bern, Switzerland, where she's now at the University of Bern. And so we caught up a little bit, and she had seen, we kind of stumbled into this zebrafish virus discovery situation. And so we might team up and look a little bit at if they're using our pipelines for sequence analysis, whether we see any interesting viruses in her data set. So yeah, this is how podcasting can kind of,
well, I think so. Yeah.
Light ideas and ripple into new. It's pretty fun.
Yeah, I think so.
Yeah. And thanks, Anthony, for reaching out. I think those are all great questions and kind of fun to think about. Okay, so I've got the next.
Yeah, it's Ben.
Next letter from Ben. Hi, Vincent and Nels. The anagrams I included in my last email were your names. Innocent Varicella, which you can scramble as Vincent Racaniello, and Cell Needs, Nels Elde. Couldn't come up with any good ones without using your middle initial.
That's nice.
Yeah, we were stumped trying to decode those anagrams last time. So thank you, Ben, for helping us out here. On a completely unrelated note, I would love to hear a discussion/paper on the gain and the loss or degradation of organelles, particularly in protozoans, whose diversity of organelles endosymbionts is truly amazing. It's currently a very welcome 25 degrees C in Adelaide, South Australia, where consistent days above 40 degrees have led to terrible brush fires across the country. All of the best for the new year. I can't wait for another year of what makes us tick.
Cool.
Thanks, Ben. Great message. And hoping for the best over there in Australia, we've been watching the news reports of all of those awful brush fires, the heat. I even saw like some relief in some, as you're saying, the temperature, also some rainstorms. But I saw this massive footage from a massive hailstorm that did all kinds of damage to greenhouses at one of the universities there. And so anyway, things seem like really have been rough.
Yeah.
For a while now in the Australian summer. It's been a tough one.
What about loss and gain of organelles?
It's a great idea. And Ben, if you have some specific papers in mind that you think we should, feel free to send those along. I agree. So I'm a massive fan of protozoans, as my PhD was devoted to one of them, a ciliate called Tetrahymena thermophila. And yeah, there's so many papers that I always have kind of my radar up to if I see a cool one. So stay tuned and let us know if there's anything that really catches your eye that you think might be worth running up a flagpole. Yeah.
All right, one more from Courtney.
Yeah,
hello from Omaha, Nebraska. It has unfortunately started to snow and is a chilly -5 Celsius. It's been -5 here the last couple of days. It's cold. Writing to tell you how excited I am that Dr. R is including an ecology lecture in his class. This was it's going to be in my virology course this year, which starts tomorrow, right. By the way.
Oh, nice.
While I consider myself a biologist, my background is in ecology, and that's the first thing I think of when I hear the term biology. However, I absolutely love molecular biology, evolution, behavior, etc. I have yet to find a subfield that I didn't wholeheartedly enjoy and want to know more about, maybe computational biology. At my school, there is a bit of a division between the molecular biologists and ecologists. A few of the molecular folks look down at ecology and don't think it's a worthy field. I think part of it has to do with there are no hard and fast rules in ecology. There's always an exception in how hard it can be to control conditions in the field, which honestly kind of confuses me. Ecology is found at literally every level of biology. Bacteria and viruses compete for space. Disturbances like antibiotics can wipe out bacterial communities in the human body, then be recolonized. The abiotic environment affects what can and cannot live in certain places. Anyways, I just wanted to express my joy hearing a molecular person incorporating ecology into their lectures.
That's great.
Yeah, I had a revelation at the Giant Virus meeting because they're all talking about ecology, basically, or most of them.
That's right.
And I just I know I should have known this because I've been listening to these folk for many years, but it just dawned on me that this needs to be in a virology course. And in fact, in 5 years, when we write the next edition of our virology book, it's going to have a chapter on ecology for sure.
That's fantastic. And also, hopefully ASV, right? American Society for Virology annual meeting, adding in some more ecology as well.
Yeah, we actually talked to someone at the Giant Virus meeting and said you should be an ecology, the ecology council, because there is someone for evolution and ecology, I believe, right?
Yeah,
and,
the next.
And he should be in it because these giant viruses really play a big role.
Yeah.
And so that's cool. I'm really, it's funny. I can see how molecular people would look down on ecologists, but ecology has changed. It's not just descriptive anymore. And I think it's gaining a lot of respect. And I think there are a lot of departments now that are, molecular biology and ecology trying to get these people together and talk to each other.
That's right. And I think it is great. And thank you for your letter, Courtney. It's really, I think honestly, this is one of the motivations of TWiEVO is to try to have those conversations and to think about, because things really are changing. And it is true just with the way science kind of came up in the last few generations and the way we specialized and the way the tools work, that it's not, when you kind of put that back, it wasn't that anyone wasn't necessarily well-meaning. It was just that it's hard to hold all of this stuff together. And now what's really fun about science moving forward these days is that now a lot of, it's not universal, but folks are starting to embrace that actually as a way forward. We've done kind of that, we're coming out of that era of specialization in some ways. And that's kind of one of the fun opportunities moving forward. And so to not only to kind of get past that old view of, well, what I'm doing is important, what you're doing, that kind of thing, but to just think, well, what's really cool and what's possible actually, and how that can kind of sustain or to provoke new progress in science. I think that's what's kind of bugging or irritating some of us in a good way to try to promote those kind of conversations and collisions in new ways.
Katie, Courtney also, sorry, go ahead, Nels. Sorry.
Oh, I was just going to say, I also agree that Courtney's point on bacteria and viruses of being a place to look, and a way of taking ecology, which is complicated, all of those exceptions, et cetera, but to try to not necessarily simplify, but just think about frameworks where it's possible to kind of consider a lot of the action that's going on in these kind of more well-described systems. And so, I think that's a good place to start.
Well, Courtney has a pick here, so let me just read this, and then we'll go on to ours.
Yeah,
I'd like to share a science pick. One of my favorite bands, Nightwish, has an album primarily dedicated to evolution/biology called Endless Forms Most Beautiful. Richard Dawkins does some speaking on the tracks. My favorite 2 songs from the album are below, with Shudder Before the Beautiful being my all-time favorite song. I cried when I saw them perform some of the songs in concert.
Oh, wow.
Endless Forms Most Beautiful and Shudder Before the Beautiful. That's cool. Did you know about this band?
No, I've never heard of Nightwish. I'll take a look.
It's very cool that they're into evolution and biology, right?
Yeah, I agree. This is a great pick. Look forward to taking a deeper dive here. Thanks, Courtney.
Nels, what do you have this week?
Yeah, I've got a pick here. This is one that just got published recently. It's called Universal Principled Review, a community-driven method to improve peer review. And so I kind of had this a little bit in the back of my mind. I was at a meeting last summer at a resort called Skamania. It was an immunology meeting, and it was a PI meeting, and kind of meant to be that way of just sort of reclaiming a meeting. So like a lot of colleagues brought their families with. We were playing like Capture the Flag. It was this great setting on the Columbia River, near the Columbia River, and just kind of mixing family and science and just ideas. And one of the things that came out of this, I didn't participate directly in this, was this idea of universal principled review. Just got published, I think, in Cell. Might be paywalled. If you're interested in looking at it and can't get at it, let us know and we'll get you a copy of the PDF. But it's kind of a set of ideas, or almost a framework, as a peer reviewer of what you might put into the review of a manuscript, for example. The reason I kind of got this back on the front of my mind was just this last week, I was surprised in a really fun way by a virologist up in Canada, Craig McCormick, had his class peer review our preprint on virus discovery in zebrafish. And so they used this universal principled review as a template to do their review. Yeah. And it was really nice. And it ended up, I think, kind of focusing on the big ideas and really kind of crystallizing the issues that are that led to a really nice piece of constructive criticism that's helping us think about as we improve the manuscript, as it's going through the peer review process in kind of out in the wild, so to speak. So anyway, I thought I would put this forward as my pick of the week. I don't know if it's perfect, but I think it at least in this one case, was sort of a nice breath of fresh air as we're considering like what are the strengths and weaknesses of this manuscript, and also a good reminder of how peer review can be really positive and kind of lift all boats in terms of improving scientific work and kind of communicating it to the larger public as well.
I think this is very cool. I'd heard some people talking about this at ASV this past summer, this idea. And glad to see that it's starting to get formalized in an article here. Transparency, I think, is really necessary. People hide behind peer review, and I think that's a big part of the problem. They say stuff they would never say to your face.
Yeah, that's right.
Right.
Yep. So they end up doing, they have all these like review categories, things like quality of the experiments, and there's a scale and kind of outlines some things, the completeness of the study, reproducibility, scholarship, again with bullet points of kind of how to think about how to break that up, the impact, which is always this kind of novelty. This is always a kind of a subjective. So these are the more subjective criteria.
Yeah.
Always challenging. Extensibility. How does this sort of reach into other fields or other, like, those kind of things? And so We've started doing this I'm teaching a class, and in fact, I'll probably sneak out of here in 5 or 10 minutes from the podcast and run over to the class. We are just finishing up a module on peer review where we gave our class 2 papers on the bioRxiv, and then had these first-year molecular biology students, biochemistry students, and neuroscience students write reviews. And we just did it sort of freeform, but I'll be handing this off to my class as a way of trying to give a little bit of a framework, or to think about some things that might help to give some form to how we do.
Cool.
Yeah.
I just want to point out Nightwish is a symphonic metal band from Finland.
Oh, no, talking.
Symphonic metal.
Yeah.
I didn't know that existed.
I haven't had a chance yet to look at these YouTube links, but I will do it ASAP. How about you, Vincent? What's your science pick of the week?
A couple of weeks ago, back in December, I was flying to Singapore, and on flights I usually read or write, or I don't usually watch something, but so it was an 18-hour flight.
Ooh, that's a long one, yeah.
So I was looking at the what they had on their videos. They had a series called Hostile Planet, which was originally made by National Geographic. You can find it on most of the streaming services have it.
Mm-hmm.
It's a series narrated by Bear Grylls. You may know his voice. If the chicks don't feed, they'll starve. But these chicks won't be able to fly for another month. That kind of stuff.
Yeah.
They had different environments. They have a series for each environment. This was some cold environment. There's a desert, there's grasslands, there's oceans. They're just really good. They're really well done. Great photography, and really interesting things going on. Of course, it's all about the impact of the climate change on animals. We all think about the impact on us, but the animals are having a lot of trouble.
Yeah, that's right.
So I found them really good. They're filmed gorgeously. The narration is minimal, so we got to keep Bear at a minimum. So they get a little bit over the top, but they do keep it to a minimum. And I really liked it. I think It's good. So you can find whatever you use for a streaming service, see if it's there. Hostile Planet. I really thought it was cool.
That's great. I'll take a look. And just getting back to Ben's letter in Australia, just some of those news reports out with, somewhere in the neighborhood of 1 billion animals.
Yeah.
Getting lost in these brush fires, these massive brush fires. So these
poor koalas, man.
Oof.
They had to kill all the wild camels because they were breaking into people's houses to drink their water.
Yeah.
I think that's really a shame.
Yep.
Oh, man. Okay. All right. That is TWiEVO 51. You can find it on any podcast player on your phone or tablet or computer. And if you listen that way, just subscribe so you get every episode. It helps us see how many people are listening. That would help us a lot.
Mm-hmm.
And over at microbe.tv/TWiEVO, you can see our show notes, links to the stuff that we talk about. If you like what we do, consider supporting us financially. You can go over to microbe.tv/contribute. There you can use PayPal or Patreon to give us a fixed amount a month, like $1 or $2, or you can give a bolus of money if you want to give us 2 years of support. Some people give us $25, $50, $100 for a year or two, whatever you want. We'll take anything because we have expenses that we want to get some help with, because we have a lot of podcasts now and there are a variety of expenses. And I'm not complaining,
Mm-hmm.
If nobody supported us, we'd still do this, but show your appreciation or something like that. Many people do. Not a lot. We get about 250 people supporting us, but
it'd be great to build that community and just give us a little more flexibility to do weird things, fun things.
Yeah, like I could go travel with Nels and interview someone, once a year. That'd be fun, right?
That's right. And if you have ideas of weird things, don't be shy about that either. We're always on the lookout for kind of capers and stuff like that. So no, it really would be great if
critter capers.
That's right. To throw some support our way. We'd both really appreciate that.
And if you have questions or comments, TWiEVO at microbe.tv. We'd love to hear from you. Don't be shy. Don't lurk.
That's right.
Someone on TWiV wrote this week, I'm a lurker, and this is the first time I'm writing.
Good to come out of the shadows from time to time.
You bet. You can find Nels Elde at cellvolution.org and over on Twitter at LEarlybird. Good to hear from you again, Nels.
Yeah, this was a fun one, Vincent. Good to be in touch again and looking forward to the next one already.
I'm Vincent Racaniello. You can find me at virology.ws. Music on TWiEVO is made by Trampled by Turtles. Man, maybe we should get what was it?
Nightwish?
Nightwish.
We'll run that up the flagpole and see if it...
I have to listen to them. I don't know what they sound like. These Trampled by Turtles are bluegrass.
They are, yeah. Good band from they hail originally from Duluth.
Maybe we should get bluegrass metal.
Now we're talking, yeah. That must exist somewhere, somehow. But we're kind of playing off the theme of, what is it? It's Turtles All the Way Down?
Yeah, that's right.
Famous thing, right?
It's a good one. Mm-hmm. You've been listening to This Week in Evolution, the podcast on the biology behind what makes us tick. Thanks for joining us. We'll be back next month. Till then, be curious.